Good afternoon. It's a real pleasure to have here Team Gollisch for the last seminar before Pesach. No more seminars until Pesach.
It looks like Matzot, no?
It is. Actually, it is recording from Matzot. We will talk about it later. So, Tim started as a physicist, and in the year 2000, he decided that he's interested in neuroscience and went to work with Idan here at the Hebrew University. And he was so excited about his work, apparently, that he continued and did his PhD in neuroscience in the group of Andreas Hertz in Berlin. And then he did, in 2004, he moved to Harvard to do a postdoc with Martisnried. And from 2007, he has been working at the Max Planck Research Institute in Munich. And this year, or last year, you moved to Göttingen. And since last year, he is a professor, full professor in Göttingen. And today, he will tell us about receptive fields. Please.
Yeah, thanks a
You
lot.
need a microphone.
Okay, thanks a lot for the introduction, Jonathan, and for this kind of invitation. It's great to be here. As Jonathan mentioned, I did my first steps in neuroscience here 11 years ago now. I have still very, very fond memories of this place. So it's always a real pleasure to be back, to see the campus, stroll around a bit, and also to hear about all the new developments that are happening here. As it always has been, it's a very dynamic and interesting place. I already had a very interesting discussions today, bridging all the way from neuroscience over game theory and philosophy to the details of the German soccer league. Afterwards, we can continue this a bit more. Today I want to talk about work that I did in recent years on the retina. I will tell you three somewhat connected short stories. One is a bit older from my postdoc work, and I will only talk about it briefly because it's already published, and then there are two new stories now from my own lab where we are continuing along the road that I started in my postdoc. It's all connected to looking at receptive fields, and these receptive fields that we look at are in the retina. This is the retina. It's a piece of neural tissue at the back of the eye, and it takes up the light. It enters the eye and processes it in this network, picking up the light by photoreceptors, and then there are a number of interneurons and finally ganglion cells that give out the output signal of the retina. These are, of course, spiked action potentials that travel to the brain. And because we have this clear distinction here between the incoming signals, the light, and the outcoming signals, the spikes from the ganglion cell, this is what makes the system so liked by many, let's say, people interested in computational neuroscience, because you have such great control over the input, and often the output is quite accessible. This is also one of the reasons that drew me into this field. So in case this has been a long day for you and you plan to nod off during the lecture, the one take-home message that you can take with you is that I want to tell you that retina is not a camera. It's not the camera of the brain, for several reasons. One is quite obvious. The output that gives in the end is not a pixel-by-pixel representation of the light that's entering the eye. Mostly because there are a lot of interconnections here between different cells and different locations in the retina. So the retina is doing something more complex, more complicated than a pixel-by-pixel representation. It is doing this more complex thing by doing interesting signal processing. And in some sense, for someone coming from physics, interesting here is almost equivalent to nonlinear, and this is one of the aspects that I will be talking about, what kind of nonlinear processing we can identify and how we can identify this in the retina. And then finally, if you look at these output patterns and these spikes here, they form, at least in my perspective, sort of intricate or interesting patterns. Their structure is interesting, and what I find most interesting here is, for example, this temporal pattern of these spike trains and the relations between different spikes, between different cells, correlations that you find there. So all these things tell you that this network is doing something complex and interesting that is more than a pixel-by-pixel representation. So why would you even think that the retina could be something like a camera? Well, this comes from, if you look at textbooks, which has a lot of useful information, but not always the complete story. But often in textbooks, what you find is that the retina is reduced to this kind of picture. There are on cells in the retina, so let's talk about ganglion cells here, the output neurons, on cells and off cells. What they do is they typically fire some spontaneous activity, and then they respond to a stimulus, for example, on cell to a spot of light by giving some action potentials. And then if the spot gets bigger, there's a surround structure of the receptive field that inhibits this response. So essentially this on cell is responding to a small spot of light and then doing some enhancement, contrast enhancement, by being suppressed by light in the surround of this small spot. The off cell is doing the opposite thing, and that seems to be a very simple linear filtering process that in the end gives you a pixel information about, or something like a pixel information about a certain spot. This is the well-known center surround receptive field structure that captures this behavior, and this is, of course, an essential part of the retina. But what I want to convince you of is that it's not everything that the retina does. There are more interesting aspects of the retina, and the retina is doing more interesting things. You can already guess by looking at its structure. For example, the fact that the cells in the retina, these 5 main cell types, from photoreceptors over horizontal cells, bipolar cells, amacrine cells to the output cells, the ganglion cells, they are not homogeneous classes. They can be subdivided in many different classes, and the most prominent diversity is here at the level where also most of the computation is happening towards the output here, these amacrine cells, which are inhibitory interneurons in the retina, and then also the ganglion cells, which are the output. They come in different classes. The current count in the primate retina is that there are 17 different classes of ganglion cells. And of these 17 cells, they all be doing different functions, so maybe there are 17 different pathways that we have to understand that emerge from the retina. Besides this diversity in cell types, there's also another aspect that's intriguing about the retina, which is the specificity in its connectivity. It was noticed already more than 100 years ago by, of course, Edmond J, who drew these beautiful drawings of the retina, where you can see that certain cells here, coming from the photoreceptors, extend their dendrites in very specific layers of the retina, and then other cells like these ganglion cells also extend their axons in specific layers, and these ganglion cells extend their dendrites in specific layers, and the different ganglion cell will go to a different layer, and therefore form very specific connections between certain interneurons and certain output neurons of the retina. In a more modern picture, this looks something like this here, in a picture from a paper from Heinz West's lab, where there's the expression of certain proteins in the retina is tagged by fluorescence markers. It's not important what these fluorescence markers actually detect, but it's important that they detect certain subclasses of cells. So, for example, the yellow cells here, in this case these are amacrine cells, and here there are yellow cells that are ganglion cells. They express both of these proteins, and therefore are specific subclasses. And you see that in the region between the amacrine cells and the ganglion cells, they have their processes in 3 very well-defined layers. So they make connections in these layers to other yellow cells, but not in the layers in between or above or below. So also the connectivity is very specific, telling us that there are probably different specific functions that are being performed by the retina. And one of the things that we try to do is identify these functions and understand how these are performed by these specific connections of these different cell types. The main technique that we do to analyze this is extracellular multi-electrode recordings. So we take a retina, we isolate it. We do this with retinas from amphibians or mouse, and the data that I will present in this talk all come from an amphibian, from salamanders in particular. And we place it, we take this isolated retina, we place it onto a dish, and in this dish there are microelectrodes, in some cases 60, in other cases then we have now 250 different electrodes, and then we can record extracellularly the spikes from ganglion cells. We also use other techniques in the lab, like intracellular recordings, but I'm not talking about this in this talk. This is a picture of a multi-electrode array. It's essentially a glass plate with these fine wires on top, and then there's a bucket in which you can hold the solution and the retina. This is a view through a microscope of a retina tissue that is sitting on this multi-electrode array. The size of these tips here, electrode tips, are approximately the size of a single ganglion cell, so that you try to, in the ideal case, have 1 cell per electrode in your recording. Then, of course, you can spike sorting to make sure that you get single units data. There's an example from a recording here, 60 electrodes with different types of spike patterns on different electrodes. For one, I would like to say typical, but in fact it's a pretty good recording channel. This looks like this, where it's very easy to identify the individual spikes. They all have the same shape in this case, or roughly the same shape apart from adaptation, so you can be quite sure that this comes from a single ganglion cell. So this is the raw data with which we work. We can use it, for example, to construct receptive fields by doing reverse correlation analyses, and this is one ensemble that we recorded simultaneously of receptive fields overlaid onto the grid of this array, just to give you an idea about the possibilities of this kind of techniques. And you can use this now to analyze the properties of the population, or just as a means to get easy or quickly get a large data sets, let's say, in a single day of many different neurons. Of course, the other side of this retinal prep is that it's easy to stimulate it precisely, and we do this like all the other visual labs by just projecting a visual display, a monitor, onto the photoreceptors by using standard optics. And the big advantage is we can get the resolution down to a single photoreceptor level, and the temporal precision of our display to whatever degree, depending on the type of projector that we take, this projector can do. So we have very high control over the stimulus. And this we make extensive use of. So the first project that I want to talk about was while I did my postdoc in the lab of Marcus Meister, and then we were trying to look at one specific potential function of the retina. And this function I named here rapid image encoding. This type of investigation was motivated by some psychophysical studies, some older ones and some newer ones, in order to give you an impression of what the kind of motivation was here. I want to do one of these psychophysical experiments with you. So what's going to happen on the screen, that behind this mask there's going to be a sequence of photographs, of natural photographs appearing very rapidly. So each photograph will be there for only a tenth of seconds, 100 milliseconds. And in this rapid sequence, you will have to detect the one image that doesn't belong to the others. And that's all the information that you're getting from the test. So pay close attention. Here we go. Here comes the sequence. Okay, I take from the reaction that you saw that one of the soccer teams was offside. So one of the players on the field was a keeper, and it doesn't belong on a soccer field. And you detected this, I hope. Most of you, or I guess most of you, detected this, although your retina only had 100 milliseconds, so in visual neuroscience terms, a short time to encode the information from this image. This is actually an old experiment that was done in the 1960s at MIT in the lab of Mary Potter, and you may know the work of Simon Thorpe in France, who has picked up on this more recently, and showed that essentially humans can do behavioral decisions based on a complex visual image like this in less than 150 milliseconds. All that we take from this finding is that you don't have much time in the retina to encode information. Therefore, we ask, how do the neurons in the retina do this? That they provide information very quickly if it's needed to come very quickly. So how do the neurons do this? The stimuli that we use in this study are not the natural images that you see here, but in some sense, let's say abstract versions of it. So we're reducing them to standard visual patterns to describe them. We use different ones of these, 8 in this particular study. It was 8 different gratings. They, at first sight, they may look identical, but they actually shifted versions of each other. So for every image, the exact boundaries between the dark and the bright stripes has slightly different locations. And we ask of the retina, can you tell us where these boundaries were exactly? So at what location the boundary between the black and white stripes are. We present them in a rapid sequence, but to facilitate the analysis, we leave a gray period in between. So the stimulus looks something like this. There's one of these in a random order, one of these grating appearing every once in a while for 150 milliseconds, and then there's a gray period in between. And then we look at what the neurons are doing, and what we find is that there is a certain class of ganglion cells that gives us a response that looks something like this. So for one type of grating, for example, for this type of grating where the ganglion cell here displayed by its receptive field, looking at the black stripe of this grating, is giving us a burst of spikes here shown in around 10 repetitions. So very reliably in each of these in each of these repetitions is giving us a burst of 3 or 4 spikes, and that's also very precise in time with a jitter of about 2 to 3 milliseconds. And the same neuron is for the opposite stimulus, for example, for the stimulus number 5 here, where T is the bright bar, is also giving us very reliably a burst of spikes, also with around 3 spikes per burst, but about 30 to 40 milliseconds later. And then for intermediate patterns, you have an intermediate timing of this burst of spikes, telling you that this neuron reacts to different examples of this set of stimuli by producing a burst of spike with a different timing. You can quantify this or visualize the quantification here by plotting, for example, a tuning curve. You plot the average first spike latency, you get a curve like this, early responses for some stimuli, late responses for others. And if you do the same thing for the spike count, you see something that is not modulated strongly, but rather flat, which means that there's sometimes more information or there's more signal information, more stimulus information here in this latency. And we believe that this is a class of neurons that does exactly this. It produces a latency code for visual images that can be read out by the brain. Not all neurons do this in the salamander retina. Here, 4 different neurons shown. First two neurons of this class that I showed you, we call them fast-off cells for historical reasons, but then there are also, let's say, more traditional textbook-like neurons like the slow-off cell or the swan cell, which respond strongly to some stimuli and weakly or not at all to other stimuli. To play the game a bit further, you can also present a natural image, and in this case we presented a natural image of a swimming salamander that is shown here. Tip of the head of the nose is here. This is an arm that is extended into the water, and then we recorded from a single neuron that was shown each one of these locations that is marked in red here. So we shifted the image so to speak over the retina and recorded, in this case, from 1,000 different locations, and recorded the spikes of these neurons. And we find for this natural image the same response as before. Sometimes it responds early, sometimes it responds late. And we can use now this temporal information to try to reconstruct this image. And this is what we get here. If we plot a dark pixel whenever there was a early response and a bright pixel whenever there was a late response, you see this gives, in some sense, a veridical or nice representation of the original image that we put in. You do the same thing for the spike count, putting a dark pixel when you get many spikes and a bright pixel when you get few spikes. You also get a representation. It looks more noisy than this representation, but it has other features. For example, this arm here is actually better represented, or the tip of the arm is better represented than here. The tip of the nose is better represented than here. So in some sense, there is more contrast enhancement in this spike count. And one of the things that we are exploring is how these two codes, so to speak, can be combined in order to give, a first pass of information about a new image, and then refine this image with more spikes that come in. But I won't talk about it here. One thing that I want to mention, because we find it interesting and find it also in many other recordings that we are doing now, is that there is an interesting structure when you look at cell pairs. Of course, looking at cell pairs is a natural thing when you think about timing, because the timing of a single cell is not well defined if there's no other information about the stimulus. Right? So in this first analysis, we measure the timing with respect to the onset of the stimulus, but the brain does not know the onset of the stimulus from other sources, or potentially doesn't know it. So the solution is to compare the timing of two cells, of two retinal ganglion cells. So here, this is the tuning curve for the first spike latency for one neuron, and here for a second neuron. And then if you take the difference, so the relative timing of the first spike from the two neurons, you see that you also get a pronounced tuning curve. Often the tuning curve in this relative timing is stronger than the individual tuning curves, in the sense that it gives you a better signal-to-noise ratio, or you can also quantify the amount of information that you get, and so often you get more information from the relative latencies than from the individual absolute latencies. One of the reasons why you get more information is, of course, because you have differential tuning. So one neuron responds early to substimuli, and the other neuron responds early to other stimuli, and then by taking the difference, you enhance, you stretch the range of responses that you get. So that's one reason for getting more information. The other reason for getting more information is that there's actually correlations, that you can find correlations between the timing of different neurons. That is shown here in a raster plot for all aim stimuli for these two cells. So every dot here corresponds to the spike timing of one cell for one stimulus versus the first spike timing of the other cell for the same stimulus. And you can see here that these clouds of spike time pairs, they are not circles. They have a elongation along in the direction of the diagonal, which means they are correlated. You can appreciate this more closely if you look at these two examples here, which means that the noise and the difference of the spike times is smaller than the noise in the absolute timing of individual cells. And this also contributes to information transmission. So this, in some sense, makes it for us a very appealing code to consider and to ask about how the retina comes about with this code. We looked a bit at the mechanisms of this code. This is yet another one of these cells that gives us this precise spike timing here and this latency relationship. I told you earlier that there are two pathways in the retina that respond to on stimuli and off stimuli, so they end up in on-type ganglion cells and off-type ganglion cells. But there's a third class of ganglion cells that's sitting in the middle, so to speak, so-called on-off ganglion cells that respond to both types, onsets and offsets of light, because they receive input from both on-bipolar cells and off-bipolar cells. And of course, this is a good candidate for these kinds of neurons, because apparently these neurons respond to anything, regardless of whether it's an increase in light intensity or a decrease in light intensity. One thing to check that is using pharmacology, for example, blocking one of these two pathways. In this case, it's the on-bipolar cells, the drug that's called APB, which blocks a certain channel that's selected for these cells, and then do the same experiment again. What you find then is that the cell goes back to be a normal, let's say, a ray-coding neuron, textbook neuron. It responds strongly to some stimuli, mainly when the cell sees something dark, and it doesn't respond when the cell sees something bright, which tells us that the convergence of these two pathways is important. And then you can go on and do a bit of modeling and use some facts about the anatomy of the retina and come up with a circuit model that we believe is doing this is producing these kinds of responses. Without going into detail, I just want to show you what the circuit model looks like and what the essential features are. So what we need in order to produce this kind of response is a ganglion cell that receives inputs from many bipolar cells. That is the case. We know it because bipolar cells are smaller than ganglion cells, have smaller receptive fields, and ganglion cells collect information from many bipolar cells. We need input from both on as well as off bipolar cells. And then we need two features. One is that the inputs from the on bipolar cells are delayed in time. They're slower than the off bipolar cells. This, we assume, comes from the different type of receptors that the bipolar cells have in getting their signal, which involves a G protein cascade, so something that's inherently slower than the typical ionotropic receptors, synaptic channels that the off bipolar cells use. So there's a reason to believe that this time delay is actually happening here in the bipolar cell. And then secondly, you need here a nonlinear synaptic transmission. And it works well if we just assume that, for example, there's a half-wave rectification happening. It's easy to understand that we need this because otherwise signals could just cancel out from on and off bipolar cells and also from different regions of the retina. So we believe that this nonlinearity is very important. And it's not the first case that this nonlinear synaptic transmission in the retina or in other systems has turned out to be, in some sense, crucial for getting, establishing a function in the neural circuit. So what we've then started in my lab is trying to look better at ways how we can analyze these kind of nonlinear transmission in the retina.
So this is before the spiking, right?
Yes, exactly. That's important. Yeah. So it's important that the nonlinearity comes before summation by the ganglion cells, so before signals could cancel, so to speak. And this is exactly what I want to address now in the second part of my talk, where we look at spatial integration in retinal ganglion cells. And this is work by a PhD student in my lab, Edmond J. And here I want to digress a little bit and, so to speak, give you a bigger picture of how we consider conceptually the approach that we use to analyze this. So I want to step away from the retina. Sorry, before I go there, I forgot about this. So we are not the first to look at nonlinearities in the retina. There are nonlinearities that have been known for many decades, and the distinction there is between retinal X cells and retinal Y cells that have been first discovered or described by Edmond J and Robson in the 1960s. And they showed that there are some cells that behave quite linearly. So, for example, you present a spatial grading, in this case a sine wave grading, on a ganglion cell, and then you reverse this grading. You go from white-black to black-white, and again, and back again, as shown here in this time course. Then for these linear cells, you find some phases of the gratings where they are excited by one switch and inhibited by the other switch. And if the grating is just positioned so that it straddles the receptor field so that the retina sees equal amounts of black and white, then you get no response because things cancel out. So the new thing in their paper was here that there are cells that cannot, where the responses cannot cancel out, that always respond. So even if the cell sees both black and white, actually as shown here, in these cases here, the receptive field of a ganglion cell just straddles the boundary of the black and white grating. They respond to both changes here, and that's a clear sign of some nonlinear stimulus integration. So this we want to look at a bit further now and try to understand what is really this nonlinearity that is doing this kind of, that's giving us these kind of nonlinear response phenomena, as well as the nonlinear integration that we need for producing this latency.
But these are ganglion cells?
These are ganglion cells, yes. So coming back to this, sort of bigger picture that I want to give you, we think of this integration as, in some sense, for us, a minimal circuit that you need for neural computation. So computation, if you think about computation at high school, it's plus and minus and multiplying and dividing. So it's always, getting two inputs and making one input out of it. And this is also, in some sense, what an essential component in neural circuits. So neurons integrate over many inputs. In the simplest case, it would be just two neurons. And what we are asking is now, can we use tools or can we identify how a neuron combines these two inputs, linearly or non-linearly, or in other words, ask what kind of arithmetic does it do on these two inputs by taking two inputs and producing a single output of its own, for example. Well, first of all, the way to analyze such a system, let's say it's a visual system, is of course to stimulate it somehow. So if you put some light intensity over the receptive field of this neuron and vary the light intensity, what you typically find is a response like this. So if you increase the intensity, the response, let's say the firing rate goes up. Maybe there's a threshold somewhere, maybe there's a situation somewhere, and you could say, well, okay, this tells me that the neuron is nonlinear. But that's, of course, not the type of nonlinearity that we want to look at. We want to look at whether the two inputs here are integrated linearly or non-linearly. So we want to distinguish this nonlinearity here at the integration stage from the intrinsic nonlinearity of the output neuron that comes, for example, from the spiking dynamics. So what you have to do is you have to stimulate each of these inputs individually, but it doesn't help if you stimulate one input and not the other. You will still get the same curve. If you stimulate the other input. What you have to do is you have to subdivide your stimulus and stimulate both inputs independently so that you can really see how they're being integrated. So now you arrive at a two-dimensional input space. So our input-output picture changes here a bit. It will look like something like this. So if you increase intensity of stimulus A, you will go up in firing rate. If you increase intensity of stimulus B, you go up in firing rate. And again, you get a nonlinear picture. But here now, because it's a model, I can tell you what the actual integration rule is here. In this case, it's just linear. I just summed A and B and then put the ganglion cell intrinsic nonlinearity, some sigmoidal function here on top of this sum. So in this simple model here, this would correspond to a linear synaptic transmission from each of these cells to the ganglion cell. You can look at a competing model like this one here, where instead of taking the linear sum, I took the sum of the squares, so this would be something like an energy model. So take A squared plus B squared. Again, I get a nonlinear response surface. And at first sight, at least these two plots seem hard to distinguish. So now we are looking for the right phase of distinguishing these two types of, conceptually different kind of integration. And the way to do it, of course, is to look at so this would be a quadratic synaptic transmission here that would achieve such a model. So the way to do it, to distinguish, is to look at the contour lines, with the lines of equal height. These are plotted down here. So in this case, because of the linear integration, you get straight lines, and here you get circles. So what we want to do now is to measure these types of contour lines in the experiments. We call them isoresponse curves, and we've actually used them earlier in the auditory system, but here now this is an application to the visual system. The question is now, how do you measure these isoresponse curves? Because essentially you always try to get the same response, so you're coming from the other end. You specify a response and want to know what stimulus you have to use to get there. So what we do to do that is we close the loop between the response that we find and the stimulus that we present by actually literally in the lab connecting the computers and have them talk to each other, and then analyze the response, for example, from a single ganglion cell, and then tell the stimulus whether it should increase or decrease its intensity, and tune the stimulus intensity until we reach the response that we want. So in some sense, this is integrated in the framework that we are interested in the lab in general, which is the framework of these closed-loop experiments, which have many other applications as well. So it requires some technological development, like spike detection, spike sorting, in an online fashion, which for the type of experiments here we can handle. It's not very difficult, and so then this is what we do. So we develop. So the detection here is very simple. We limit ourselves to those electrodes that give us a really good signal-to-noise ratio at this stage, where we can detect single spikes just by thresholding them in the exocellular recording. And then we use this, for example, to find the receptive field of the cell. Also, we do this by a simple online algorithm. These are all off cells that we are analyzing, so they respond to a darkening of the stimulus space. And what we do is we subdivide the stimulus that we give into two halves, and then we present the stimulus on the left and the right, with something like this. First on the left, then on the right. We count the number of spikes and compare spikes to stimulus on the left, spikes to stimulus on the right. And if there are, for example, more spikes on the right, we just shift the boundary to the right, and then do it again until we have tuned in so that we get equal amounts of spikes from left and right. So that gives us the middle of the receptive field in one direction. X direction, we do the same thing in Y direction. And we have the two-dimensional center of the receptive field, and then we just take spots of different sizes and find the maximum response for these types of spots that give us the size of the receptive field. So after this initial tuning, within a few minutes we have an outline of the receptive field of a single ganglion cell. And in this receptive field now, we can present a stimulus, as I showed you before, which has two components, one on the left, one on the right, and we can individually put different stimuli here and look at how the cells integrate these two visual contrasts. So what we do then in order to find a specific response is first we decide on a specific response. Let's say for a 500 millisecond presentation of the stimulus, we want 4 spikes on average from the stimulus. So what we do is we choose a ratio of these intensities here on the left and the right. And then we fix that ratio and co-vary them by going up and down until we find the right response. We start at low contrast, which is important because if you take too high of a contrast, you easily run into a scenario where the retina is strongly adapted and you have to wait for a long time. So this is another benefit of doing this with online analysis. You can avoid regions where you run into the wrong adaptation regime. So we start at low contrast, we ramp up the contrast until we find the region of interest here, for example, for 6 spikes. And then we do a little search algorithm in this region, a greedy algorithm that then tunes in onto the right, as good as it can, onto the right intensity that we need to get our 6 spikes. We do this for spike count, and we also do this for latency. For example, in this case, finding a latency of 80 milliseconds. And then we can compare the curves that we get. So this will give us now a single point that gives 6 spikes, but then if we change the ratio of our two stimulus components here, we search in a different direction of stimulus space and get another point. And we do this a number of times until we get a whole ensemble of points, and then we look at the shift. So the space in which I'm presenting These isoresponse stimuli looks like this. So we are looking at off cells, which means they are responding to negative contrast. So we also always start at the mean gray level, and then, for example, decrease light intensities, which corresponds to a negative contrast. If I would go on the diagonal to the lower left, this would be a homogeneous stimulus, equal contrast on left and right. If I go along the axis, for example, only one of these stimulus components is used, and the other one stays gray. In this space, I'm going to be looking at the responses. So a typical example response looks like this. This is from a single ganglion cell where we measured both an iso rate curve and an iso latency curve. And the first thing to observe here is that these curves are nonlinear. So the ganglion cell is not doing the simple thing of linear integration, which is sometimes we expected because from these earlier experiments we knew that there are a lot of nonlinearities in retina. And this is what we also see here. And then we can analyze these the nonlinearities that make these shapes a bit further. For example, one thing that one realizes is that in these quadrants here, where one contrast is positive and the other is negative, these curves almost run parallel to the axis, which means they completely ignore how much positive contrast there is. They are only affected by the negative contrast. So this really is a sign of half-wave rectification. Everything. So the non-preferred stimulus, in this case, is the positive contrast, is just thresholded away. And then in this region here, where there are two positive contrasts, this looks more or less circular. So also here it's not a straight line. It's more circular or elliptical, which means that there's something like a quadratic integration. We can actually use this to fit a model where there are, for example, two populations or two inputs feeding into a ganglion cell, and then there's a nonlinearity here at these synapses, and these are then the fitted synaptic transmissions that corresponds to these curves. It's maybe what you would expect. There's a strong rectification, and then there's a gradual increase in the gain of these neurons. So something we call it a threshold quadratic integration, or threshold quadratic nonlinearity. This is what we typically find. Here's another example where the rectification is not as pronounced, and it's going a bit towards the more linear side of this integration here. So this is about the range of integration that we typically find. And now we could say, okay, the job is done. These are the nonlinearities that we find in the retina. So now we know how the retina integrates visual contrast over space, and we'll be fine for that.
So this inhibition, negative parts of the spatial, no, the negative part in your curve, the nonlinearity.
Here?
Is mediated by some inhibition?
So probably not by inhibition, because if we use inhibition blockers, this doesn't change much. So it's probably just imperfect rectification of the bipolar cell. So which means there should be a steady efflux of neurotransmitter, and then if the bipolar cell is suppressed, so to speak, this efflux is still reduced. So it can go down a little bit, but it can much more easily can go up. So maybe fortunate for us, or interestingly for the retina, we also find some cells that are doing something very different, and they produce iso response curves that look like this. So the iso latency curves here look very much like they always do in these cases, rectified and then quadratically integrated. But the iso rate curves are doing something very funky. They have a sensitivity maximum, so you need very little contrast here for homogeneous stimuli, so along this diagonal. So there seems to be a particular sensitivity to this kind of stimuli where you need very little contrast to drive the cell to, in this case, the H spikes that we were looking for. And this is something that pertains to the rate, but not to the latency for these cells. So they are integrating latency and rate differently here. This is what I want to look at a bit further now. We call these cells homogeneity detectors because as long as you look at the rate, they're very sensitive to homogeneous stimulation. This is what this curve tells you.
Can you change the number of spikes? There?
Good question. Doesn't have a big effect.
That's good.
It doesn't have a big effect. So the type of cells, whether it has this kind of non-convex shape or whether it has this kind of threshold quadratic shape, remains the same. Some details change we can attribute to adaptation. But the general pattern stays the same. Unfortunately, I don't have any data here to show you this. Okay, so one thing that we want to do is we want to understand on what spatial scale these nonlinearities happen, both for these normal cells as well as for these homogeneity detectors. This we can do by a little variation of the stimulus. Instead of separating it into two halves, we now interleave the contrast. For example, by putting the contrast patterns into such a checkerboard pattern, and making the checkers smaller and smaller, and then seeing, you still get the nonlinear integration if the two contrasts are strongly intermixed on a small spatial scale. And of course, we see that eventually these nonlinearities break down. So if you make the checkers smaller and smaller, these curves become more and more linear, which is expected. At least, once you're below the resolution of a photoreceptor, we know photoreceptors are linear. This curve has to be linear. The question is, when is this happening? This is true for these normal cells, as you see here, the curves linearize, as well as for these homogeneity detectors. So both this nonlinearity, this hypersensitivity here to homogeneous stimulation, as well as the rectification disappear if you go to fine enough spatial scales. You can quantify this by looking, by using different types of these patterns and looking at what spatial scale this breaks down. If you fit this, so this is the slope of the rectification here. 1 would mean there's no rectification, 0 would mean there's perfect rectification here in these quadrants. So you see a dependence on the size of the stimulus pattern, and the critical point of the Finland exponential. The scale is around 100 micrometers, and that's approximately the size that we know bipolar cells have. So this is for us a strong indication that we are really looking at the right model here, at a model where these nonlinearities happen at the bipolar cell level, and not, for example, at the photoreceptor level, which are much smaller, which would imply a much smaller spatial scale. So we can look at this type of model a bit more seriously. So then the question is, well, how do we get this particular weird sensitivity to homogeneous stimuli? So now that we have these iso-response curves, we can start looking at them in more detail and asking, for example, what is the actual spike pattern? How do the spike bursts look like at these locations or at other locations? So this is what we do. We put ourselves onto different points of such an iso-response curve here for a normal cell, and then we repeat these stimuli here, these characteristic stimuli, many times, so that we can measure, for example, a PSTH, a stimulus time histogram. And for this normal cell, not surprisingly, the bursts are virtually indistinguishable whether you sit here in the region where there's a homogeneous stimulus, here spikes shown in black, or whether there's only 1/2 of the receptor field is stimulated here in green or in orange. Spike patterns are very similar to PSTH. This is, of course, not the case if you look at one of these homogeneity detectors. There you find that for the stimulus that gives you the desired number of spikes for homogeneous stimulation, shown here, these spikes come much later than we know from the iso-agency curve, but also more spread out in time. So this, the time, the interval during which the cell response is longer than for these earlier responses when only 1/2 of it. So there's a characteristic difference in the spike patterns, in the shapes of the responses. You can see this also when you look at spike patterns that come from an iso-latency curve, so from stimuli that give you the same latency. You see this here. The homogeneous stimulation, of course, gives you more spikes now because you're outside of this iso-ray curve. It gives you more spikes, but with the same latency. And both of these pictures somehow suggest that for these stimuli here, when only 1/2 of the receptive field is stimulated, the response is somehow cut off here at the end. It's truncated in time. It's sharper here, it's sharper here. The spikes that seem to be missing here are the ones, the trailing spikes at the end. So this tells us that probably the mechanism that we have to look at is some sort of dynamic gain control and dynamic adaptation that is cutting off the spikes for one type of stimulation, for the stimulating only 1/2 of the receptive field, but it is not effective for a homogeneous stimulation. So this is what we're looking at. There are some easy candidates. One easy candidate is synaptic depression. That's easy to see. You don't have to build a big model. The idea is here just if you stimulate only 1/2 of the receptive field, the synapses in this 1/2 have to work much more in order to give the same excitation than if you distribute the load over many synapses as you can do in the homogeneous case. And therefore, there's a possibility that synaptic depression will be more effective here for these stimuli, and then the neuron tires earlier, or the input tires earlier. So we tested for such a mechanism by trying to pre-adapt the neuron, or pre-depress the synapses by presenting here a short stimulus that precedes our actual stimulus that has two components. And we present the stimulus only on one half, so we're trying to pre-adapt the synapses on one half of the receptor here and see what effect this has on our iso response curves. And the hypothesis is that we should see a much stronger effect for these homogeneity detectors. If this is the mechanism that makes the homogeneity detectors be homogeneity detectors. And to cut this short, what we find is that for the normal cells, we see that there's an effect of this pre-depression, because this iso response curve here shown in green compared to the control in blue, it moves outside, and as you lose sensitivity. And it moves to the outside in an asymmetric fashion. It moves further to the outside for the stimuli that are on the same side where you did the pre-depression. So you lose sensitivity more on the side where you stimulate, which is exactly what you would expect. And this would be the fact that you have stronger effect on one side than on the other side. That's a sign of a local adaptation or local gain control mechanism. We see that this is the case here for the normal cell. We also see that this is the case for the homogeneity detector, but by no means it's much stronger here than it is here. On the contrary, often it is weaker for these cells than it is for these cells. So all these cells seem to have a local adaptation mechanism, which could be synaptic depression, but it's not stronger for these homogeneity detectors. So it doesn't really look like that this is the mechanism that makes these cells be so sensitive to homogeneous. The alternative hypothesis would be that there is a mechanism that is mediated by inhibition. So there the idea would be that you have a local inhibitory circuit that controls the gain at this location. And the easy way to test any effect of inhibition is to put inhibition blockers onto your prep and see whether anything changes. And what we find there is that indeed things change very dramatically. So here is again from such a homogeneity detector an iso rain curve shown in blue. And in orange, this is what you get if you put inhibitory blockers on there. So first of all, the ganglion cell becomes much, much more sensitive because inhibition is lacking. So this iso response curve moves to the inside. You need less contrast to get the same response. But also you see that the shape changes. It now looks more like an iso response curve from the normal cells that we had before. It's now convex again. It shows the rectification still, and it's almost quadratic here in the central region. So this is the first hint that inhibition is involved here. Now we could of course say, well, you're changing the way that you activate your synapses. Apparently you need much less contrast now. So maybe this is the reason, because you need less contrast, you're not activating the whole system as strongly as you did before, and therefore you're not triggering the mechanism that makes the cell here give it this shape here as the blue case. So as a control experiment, what you can do is here you can try to reduce the effectiveness of your stimulus here in the case when you give inhibition blockers by making it smaller in space. So then in the end that every synapse has to work again the same amount that it used to work in the case when there was inhibition. This is shown here, so if you make the stimulus smaller here as shown for the screen curve, as you would expect, this iso response curve again moves to the outside. You need of course more contrast now to get it back to the normal response. But it doesn't revert the changes that you see in the shape. It still remains convex as it did here. So this really is for us the indication that this strange sensitivity to homogeneous stimuli here comes from some inhibitory interaction in the network. And we can actually model this, a very simple model by just saying that there's some inhibitory, some local inhibitory either feedback onto the bipolar cells or feed forward onto the ganglion cells. That doesn't matter. Important is that it's local, that it takes a little bit of time because it doesn't affect the first spike, but only later trailing spikes, which can be explained by the fact that you have an additional synapse here, so it's natural that it comes with a certain delay, it's slow, and it has to be high threshold, or more strongly nonlinear than the bipolar cells have to be, because only at strong contrast levels it really is being activated. And this is then the mechanism that can explain this. If you use only one half of your receptor field, again you're very strongly activating one of your two pathways, and that inhibition is recruited. if you distribute the load on both or all of the pathways in your receptive field, and the local activation is not so strong, and inhibition stays below threshold, so to speak. So that's the mechanism that we imagine. Of course, it's then interesting to ask what function may this serve. It's easy to imagine what you may need cells for that detect homogeneous stimuli, namely to detect a homogeneous stimulus, which could be something important, like, for example, a predator that is approaching you. You think of an amphibian, for example. So if you take such a virtual visual environment where there's a dim homogeneous object and then high frequency noise or clutter here, and then you work through different models of spatial integration. You can, for example, start with a linear model where you say, okay, I'm dividing the space now into different squares, and each square which corresponds to a ganglion cell, and this ganglion cell integrates the stimulus over the stimulus that falls into its receptive field. Then this linear integrator, which gives you linear iso response curves, detects both of these types of stimuli because they are on average have similar contrast. If you use the normal cell here that has this threshold quadratic integration and this convex iso response curve, this is very sensitive to this high frequency clutter essentially because of this nonlinearity. So it's good for detecting small stimuli. If you look at a cell that may be one of these homogeneity detectors with a high sensitivity to homogeneous stimuli, which you can actually model by this type of synaptic transmission, just in a simplified model, then you see that this cell now, or the array of ganglion cells with these types of cells, are very insensitive to this clutter and very sensitive to the homogeneous stimulus as you would imagine. And therefore it may be useful for a retina to have these two pathways to separate information. Okay, I would have another 5 minutes story, and I will briefly go to the last part, which really is brand new. So if you have any input into this afterwards, I would very much appreciate it. Where we ask these subfields that we have so far analyzed, how do they respond to adaptation? Essentially, we're asking a question about contrast adaptation, and we're asking whether it's a local property. For example, do the subfields adapt independently, or is the global property of the whole ganglion cell, so it's something that the ganglion cell does in its entirety. The reason why we're interested in contrast adaptation is because contrast adaptation is very prominent in the retina. The way it's analyzed typically is that you have a flickering sequence of light intensity, shown here, so it's different gray values of your stimulus screen, and you go from low contrast to high contrast and back. You see that the cell responds better to high contrast, you get more spikes. But what people typically analyze, or what's interesting here, is the fact that the integration characteristics change. And this is modeled by a model that consists of a linear filter and a nonlinearity. And without going into detail, this filter here describes the integration kinetics of the neuron. So you can think of this as the preferred stimulus of the neuron, or if you look at it the other way around, as the impulse response function of the neuron. So what's important here, only for our purpose, is that in the high contrast condition, this filter looks different. It's faster, it has an earlier time to peak here than in the low contrast condition where the green filter is relevant. And this is one adaptation property. The other adaptation property is the change in sensitivity, which is measured by the input-output relationship. You can think of this as there's here in the green curve, the output light is earlier, there's a lower threshold, and therefore at low contrast the cell is more sensitive. So these shifts here in the nonlinearity multiply the sensitivity of it. And now we want to ask whether these properties here in the response kinetics as well as the sensitivity are local or global properties. So what we do here is we change the stimulus paradigm and go from this homogeneous stimulus to an inhomogeneous stimulus, where we have one location that we call X, one location that we call Y, spread out over the stimulus display. And here they are overlaid with a measurement of the receptive field of the ganglion cell. There is this colored patch here. This is just to show that a typical ganglion cell sees both stimulus X and stimulus Y in multiple copies. This is just a screenshot, so there's a gray background, and then at this In this case, X is just having a bright intensity and Y is having a dark intensity. But both of these intensities are randomly flickering in time, just as we had here, high contrast and low contrast. And now we use a stimulus that changes only at one of these locations. At location X, it changes between low contrast and high contrast, and at location Y, it always has the same contrast. And what we can do now is we can use this model to get the filter that describes the kinetics only for the location X, and we can get another filter only for the location Y. The reason that we can do is because these stimuli are independently drawn from each other. So the question now is what happens to these filters and non-linearities in X and in Y. And keep in mind that now at location Y nothing's happening. So if there's so you may imagine nothing should change for location Y, at least if adaptation is local. So what we find here is, for example, for a neuron for location X, the filters change as you would expect. So if contrast is high at location X, this is here shown in red, the filter is again faster than during the time when its contrast is low at this location, as we had it before. Now the question is what's happening at location Y during these two phases. And what we find there is that there is a very similar change in the filter kinetics. So also here in red, the filter at location Y is faster, when at location Y nothing is happening, but at the other location there's a higher contrast. So in some sense, the ganglion cell seems to spread its adaptation properties over its whole receptive field. It's affecting stimulus integration at any location in its receptive
So you
field.
measure this by probe at Y?
Sorry?
You measure this by adding a probe at Y?
No, I mean the stimulus is the probe itself. So the stimulus.
But it's not at Y.
So at Y, the stimulus is also flickering. So this is in some sense the envelope of the flicker. So the envelope is staying constant, but within this envelope, it's flickering just like here, or what we actually use is here. So you can imagine the stimulus at Y is a sequence like here, but without these, extended amplitudes here.
These are the same flickers?
No, they're independent flickers at X and Y. And at Y, the flicker always looks like this, so it has this amplitude, and at X, it's changing from this amplitude to this amplitude.
What's the spatial scale here?
So the spatial scale we try to match here to bipolar cells. So each of these checkers here is around 80 microns on the retina. And then we leave some space in between because we want to minimize the number of bipolar cells that see both. So that we can, as best as we can from the anatomy that we know, separate the input streams that go into the ganglion. So to wrap this up quickly now, we did a control experiment where we now also change the contrast at Y, but in counter phase. And we use this as an example where the total contrast always stays constant, but it's differently distributed between X and Y. What we see then is that essentially the kinetics of these filters do not change. So the peaks here between the blue and the red curves are at the same time. So again, the contrast adaptation seems to be global, because global contrast is not changing, the cell has the same kinetics. There is a local change, however, that becomes now apparent in these kinds of stimuli. So here, the blue curve is shifted upward, or it's becoming more biphasic. The cell is becoming more high pass, if you look at it from an engineering view. And here, at this location, the red filter is making the cell be more high pass, more biphasic.
It also creeps here.
Exactly. So this is consistent with the fact that here the red curve is shifted upward or becoming more biphasic. So this essentially is a point that we are now working on, which is quite puzzling. So most of the effects that we see are global changes in contrast adaptation, but then there is this shifting upwards, so this changing in the biphasicness of these filters, and the integration properties making the cells more high pass. That is happening actually at the location where the contrast does not change. So the interpretation here that this is really an effect of the surround. If we look at location Y, the surround of this location is made up by location X. So the surround really is giving us a contrast adaptation effect that changes a different property. This is then something that at least in the retina field is new, that there are two distinct mechanisms of contrast adaptation, two different things that are happening, one apparently at a global scale and the other at a local scale within the ganglion cell receptor field. save some time, let me just say that for the nonlinearities, we also find a global mechanism. So here you see that at location Y, although contrast is not changing here, the red curve is below the blue curve, so sensitivity is reduced. Here, also for stimulus integration at location Y, when contrast is higher at location X, which means that the change of sensitivity also seems to be a global property of the ganglion cell, giving us also some ideas of what the potential mechanisms for this is. So the current hypothesis is that's probably inactivation of sodium channels with spike generation mechanism. And then, very last, what could this be, or why is it interesting at all to distinguish between local and global adaptation? And for example, if you had a local adaptation mechanism, you could argue that this is useful because if you have stimulation at one part of your receptor field, for repeated time you adapt to it, but you stay sensitive to stimulation at another part of your receptor field. So you retain, if you want, the sensitivity to a novel stimulus, even if it falls onto the same ganglion cell. But this is not what we observe here for the retina. We observe global adaptation, but there's also a paradigm or something that has been looked at for which global adaptation would be very useful. And this is, for example, if you have a stimulus that moves into the receptor field. So it starts moving into the receptor field from one side of the receptor field, so the ganglion cell already sees that the stimulus comes, and then starts adapting, and then when it moves to the other side of the receptor field, the ganglion cell is already adapted and giving less of a response. So the ganglion cell is more sensitive to the entry of a stimulus into the receptor field than to the exit, so to speak. This has been related to a phenomenon that is known as motion anticipation, and it's thought to cancel the delay that you have in your neural circuit by having to first detect the light and integrate the light, but in order to detect a stimulus, a moving stimulus, at the location where it is now, you want to cancel the delay somehow. And one possible mechanism for canceling this delay would be to be more sensitive to the entry, and then you say, I saw something, and the readout mechanism can say, okay, this guy saw something, so I'm assuming that there's a stimulus here, and this is where the stimulus will actually move to. So you detect it at the right location. This has been proposed to happen in the retina by a paper in the Martisnried lab. They call it motion anticipation, but it really is this cancellation of transmission delay. For this, you need global adaptation, which now we can show.
This is velocity dependent?
This is velocity dependent. So there's an optimal velocity for which you can anticipate. If you had multiple streams, then you may be able to do this for multiple velocities. Okay, let me quickly sum up. First part, I talked about the fact that we observe in some ganglion cells a latency code. Cells respond for some stimuli earlier than for others. And we could explain this by this type of nonlinear circuit model, where in particular a time delay of this one input time and this nonlinear synaptic transmissions were important. Then we looked at more closely these nonlinear synaptic transmissions by a technique that we propose of measuring directly these iso-response curves, these contour lines. We found different kinds of more or less interesting nonlinearities in the ganglion cells that we think are worth exploring further. And then finally, we briefly looked at contrast adaptation, especially at the spatial extent of contrast adaptation, and found that at least in the type of experiments that we do, most adaptation effects seem to be a global property of the receptor field, apart from this weird shift in the filters that somewhat change local integration characteristics. And then let me just end by thanking my group. This is my group, as I'm sure was assembled in Munich. We're now moving to Göttingen. Some of these guys are coming along. And then the first part of the story was done while I was in Martisnried. Thanks a lot.
Question?
So, maybe I missed it, but you haven't told us about how the nonlinear model generates the data.
Yeah, so I skipped this a little bit. Well, the short answer is that it does. The longer answer is that if you think a bit about it, it's trivial. Essentially, it means that because of this rectification here, essentially you from every location you get excitation. If at this a certain location you get white, then this on pathway gives you excitation, and at this location the stimulus turns black, this off pathway gives you excitation. So that in the end, you always end up with the same amount of total excitation if you want to. This is why we believe in the end we always see, for example, 3 spikes as we saw for this one particular neuron. So the total amount of excitation is the same. But the timing of this excitation depends on whether the excitation is provided by off ganglion cells, which are faster, off bipolar cells, which are faster, or on bipolar cells, which are slower. So if all the excitation comes from on, it's the pure brightening of the stimulus, then it just takes more time for this excitation to arrive, and you get a later response. For black, you get an earlier response because you draw on these fast off bipolar cells. And then for stimulus in between, you get something in between.
And the time scales match what we know about the slowness of the bipolar cells?
As far as we can measure it with the time scales match. So what we can do, we can compute a spatial temporal receptive field separately for the on inputs of the ganglion cells and for the off inputs of the ganglion cells. Now, if we make the assumption that the ganglion cell itself is treating its on inputs and its off inputs the same way, so that it's not, because, I don't know, local dendritic processes do anything else, which would probably in the end be equivalent. But if we assume that the ganglion cell is treating the same things, treating its on inputs and its off inputs in the same way, then we can look at the time scales of these spatial temporal filters of the on and off input separately and compare that time. So there we indeed find a 30 to 40 millisecond difference in these spatial temporal receptive fields, and this is what we think underlies this different times here for a completely different stimulus. So that matches. We actually use that to model these outputs. So we use the spatial temporal receptive fields to model these outputs, and it's a very good match.
You talked about 3 typical bipolar synapses, right? Of these 2 of the shapes here and another one. Can you say something about the distribution over the retina? Because we can measure that, right?
Yeah, in principle we could measure it. Unfortunately here we cannot really measure it because we really only analyze a single cell at a time. So we are not we're using the fact that we have many electrodes in this experiment only to select the one electrode that gives us really good signal, in disregard to 59 other electrodes. Because then we optimize the stimuli for this one cell and measure on this one cell for 2 hours or 3 hours, and then maybe we'll go to the second cell, but we don't look at the full population with these characteristics. So therefore, I can't say anything about a particular distribution. I can only say that in the salamander retina, we observe this kind of cell about 30% of the time. Although that does not mean that it exists 30% of the time. I mean, we probably have a sampling bias here because we're restricting ourselves. First of all, we're restricting ourselves to cells that gives big spikes. So this is the limitation.
But related to it, you started with showing us the beautiful anatomy and specificity of. So when you say 30% here, or if you look about the global adaptation, can you relate it to the specific types of cells?
A little bit, yes. So the anatomy that I showed you was unfortunately not from the salamander, but from the rabbits. So there the anatomy is quite clear. In the salamander, it's much more messy. The one thing that we can do is we can relate it to other properties of the receptive fields, for example. So what we find is that we get these types of cells exclusively if the cells have a very big receptive field and have virtually no surrounds of the receptive field. This relates actually to other studies that people have done in frog retinal ganglion cells, where they proposed that there are cells that have no surround and that they call them limbing detectors, and they have actually already hinted or proposed that these cells are specific for the escape response of the frog. So, for example, if you inject inhibition blocker into the eye of the frog and they give it a stimulus to which it would normally jump away from, it doesn't do it anymore. Which goes is somewhat in line with what we found, that these cells are strongly affected by inhibition. The inhibition makes the cells be the types of cells that they are. and they have the same properties as these cells in the earlier stuff. But that's as far as we can go.
And these folk experiments, they respond to other stimuli?
Sorry?
They still respond to other stimuli?
The frogs, you mean?
Yeah,
They can catch fly.
Yeah.
Good question. I would have to go back to the paper. I remember that there is a control experiment that the frogs still see, but I don't remember what exactly they tested there.
Maybe they injected it to the legs rather than... More questions? So I have one question. So in the model you have all these nice ESO response curves, but in your experiments you only showed us like one ESO response curve. Is it just because you only measured one, or for the sake of presentation?
A little bit both. So we often measure only one curve because then we want to do other measurements, let's say inhibition blockers or measuring PSAHs, but of course we have done control experiments measuring multiple curves. This question was already earlier. If we go, for example, to a different spike count, then essentially we see the same shape. So that's not dependent on the spike count. There's subtle differences between the curves which are well worth analyzing because we can see traces of adaptation there. But overall, so the classification is independent of many stimulus parameters, whether we make the stimulus big or small, look for 8 spikes, look for 4 spikes, so this seems to be independent of these structures. But next time I will put a picture in at least 2 of these guys, because we have it.
2 is many. More questions?
For experimental stuff.
Let's thank our speaker again.